swallow. That's it. That's the last voluntary thing you're going to do to the meal you just ate. From here on out, about 20 feet of tubing takes over the job, and none of it is going to ask your permission first. Right now, somewhere behind your navel, there's a tube about the width of your thumb, contracting in a rhythm you have never once felt, mixing what you ate with a chemical cocktail so precise that being off by a few minutes can be the difference between your body absorbing that meal or losing it entirely. This is the small intestine, and small is a little misleading. Uncoiled, it's about 7 meters long in an average adult, roughly the length of a school bus. It's small in diameter, not length. Over the 4 to 6 hours after you eat, it runs one of the most tightly choreographed chemical operations anywhere in the human body. Taking a sandwich or a bowl of rice and breaking it down into molecules small enough to slip across a single layer of cells and into your bloodstream. You haven't felt any of this happening. You're about to understand all of it. By the time food reaches the small intestine, it's already been through the ringer. The stomach spends 1 to 2 hours churning it with hydrochloric acid at a pH somewhere between 1.5 and 3.5, plus an enzyme called pepsin that starts hacking proteins into rough pieces. What comes out the other end is called chyme, a thick, acidic slurry that's no longer solid food, but isn't yet anything your body can actually use. That slurry crosses through the pyloric sphincter into the first stretch of small intestine, the duodenum, and it doesn't arrive all at once. It comes in small squirts, 2 or 3 milliliters at a time, a metered drip, not a flood. And the reason for that metering is the first mechanism worth slowing down for, because the duodenum simply cannot handle what the stomach just handed it. That chyme is acidic enough to damage the lining of the small intestine, which isn't armored the way the stomach is, with its thick coat of protective mucus. So the instant that acidic chyme touches the duodenal wall, cells there release a hormone called secretin into the blood. This, historically, was the very first hormone anyone ever discovered, identified back in 1902 by William Bayliss and Ernest Starling at University College London. They found that a strip of intestine with every nerve severed still made the pancreas respond to acid, which meant a chemical messenger, not a nerve signal, was carrying the instruction through the bloodstream. Secretin travels to the pancreas and tells it to dump bicarbonate, yes, the same stuff in baking soda, straight into the duodenum through the pancreatic duct. Within minutes, the pH climbs from around 2 up toward neutral, close to 7. The burning acid the stomach worked so hard to build gets switched off almost instantly because everything about to happen next depends on a neutral environment. The enzymes waiting in the wings simply don't function in acid. They denature, they die on contact. Once the acid's neutralized, a second hormone shows up, cholecystokinin, or CCK, released by a different set of cells in that same duodenal lining, and it's triggered specifically by fat and protein fragments in the chyme. CCK does 2 jobs at once. It tells the gallbladder to squeeze, releasing stored bile, a greenish-yellow fluid the liver's been manufacturing non-stop, held in reserve in the gallbladder for exactly this moment. And it tells the pancreas to release its whole enzymatic arsenal, amylase for carbs, lipase for fats, and a group of proteases, trypsin, chymotrypsin, for protein. All of that because a stretch of tube about 25 centimeters long chemically sensed what you ate and gave orders to 2 separate organs in response. Bile itself isn't an enzyme. It doesn't break a single chemical bond. What it does is closer to what dish soap does to a greasy pan. Fat entering the intestine tends to clump into big globules, and big globules have very little surface area relative to their volume, which means lipase, the enzyme that's supposed to digest fat, can only reach the outside surface. Against a big blob of fat, it's basically useless. Bile salts solve that. They're amphipathic. One end loves water, the other end loves fat, and they wedge themselves into those globules and shatter them into a fine emulsion, droplets about 1 micrometer across. That single move multiplies the total surface area of the fat by a factor of thousands. Now lipase is working on a mist instead of a boulder, and it can finally clip triglycerides down into free fatty acids and monoglycerides fast enough for it to matter. While all of that's happening, the muscle wall of the small intestine is doing something you'd never guess from the outside, from just feeling full. It's not simply shoving food along. For the first several hours after a meal, the dominant motion is something called segmentation. Localized ring-like contractions popping up every few centimeters along the tube, squeezing the chyme forward and backward, chopping it, folding it, working it over. More like kneading dough than pushing something through a hose. This mixing pattern, first mapped out systematically through manometry studies in the mid-20th century, makes sure every particle of food gets exposed to the digestive fluids and, eventually, to the surface that actually absorbs it. Only later, 4 to 6 hours in, once most of the nutrients are already gone, does the intestine switch into what's called the migrating motor complex. A genuine forward-sweeping wave that clears out whatever's left. That's the housekeeping cycle. It's the sound you sometimes hear as stomach growling between meals. And that sound isn't hunger knocking. It's the intestine confirming there's nothing left to mix. By this point, proteins are being cut down into short chains and single amino acids by trypsin and chymotrypsin. Enzymes the pancreas is careful to manufacture and store in an inactive form, trypsinogen and chymotrypsinogen, specifically so they can't turn around and start digesting the pancreas itself. Only once they reach the duodenum does an enzyme called enteropeptidase, sitting right there on the intestinal wall, cleave them into their active forms. Get that switch wrong, and the pancreas starts digesting its own tissue, which is essentially what acute pancreatitis is. The body builds in physical distance and a molecular safety catch between where the enzyme is made and where it's actually allowed to become dangerous. Carbohydrates go through their own version of this. Salivary and pancreatic amylase break starches down into shorter sugar chains and eventually into maltose. But the final cut, turning maltose, sucrose, and lactose into single molecules of glucose, fructose, and galactose, doesn't happen out in the intestinal fluid at all. It happens right on the surface of the absorbing cells themselves, on a structure called the brush border. And this is where geometry becomes the real hero of the story. The inner wall of the small intestine isn't smooth. It's folded into circular ridges called plici circularis, and those folds are covered in finger-like projections called villi, roughly 1 millimeter tall, packed so densely they give the tissue a kind of velvety texture. And every single cell on the surface of every villus is itself covered in thousands of even smaller projections, called microvilli, which under a microscope form what's known as the brush border. Folds on projections, each layer multiplies the surface area by another order of magnitude. For decades, textbooks quoted a staggering figure that the small intestine's absorptive surface was roughly the size of a tennis court, somewhere around 200 to 250 square meters. Then in 2014, Herbert Helander and Bjorn Fendrix at the University of Gothenburg went back and remeasured it properly, accounting for the fact that villi flatten and stretch when the intestine is full of food, and revised that number way down to about 30 to 40 square meters. Still roughly the size of a small studio apartment, folded up inside a tube you could wrap one hand around. That correction didn't make the intestine less remarkable. If anything, it made the number honest, which is its own kind of remarkable, given it took over 60 years for anyone to actually go back and check. It's on that brush border, on the microvilli themselves, that the final enzymatic cuts happen. Lactase, sucrase, maltase, physically anchored right to the cell membrane, so the moment a sugar molecule gets split apart, it's already touching the doorway into the cell. And that doorway isn't just some passive gap. Glucose gets pulled into the cell by a specific transport protein called SGLT1, the sodium glucose cotransporter, which uses the cell's sodium gradient, kept up by pumps burning ATP around the clock, to drag glucose in against its own concentration gradient. It's the exact same molecular trick behind oral rehydration therapy, that simple mix of salt, sugar, and water the World Health Organization credits with saving tens of millions of lives from diarrheal dehydration. Because sodium and glucose delivered together get absorbed even when the gut is otherwise in crisis. Once inside the cell, glucose exits out the other side into the bloodstream through a different transporter, GLUT2, and gets swept away through the portal vein straight to the liver before it ever reaches the rest of your body. Fat takes a completely different path, and this is the layer most people have genuinely never heard explained. The fatty acids and monoglycerides that bile emulsified and lipase clipped free don't dissolve well in water, So they travel through the watery intestinal fluid packaged inside structures called micelles, again, built out of bile salts, ferrying the fat molecules over to the absorbing cells. The way a delivery truck hauls cargo through terrain, it could never survive crossing on foot, unprotected. Once inside the intestinal cell, the fatty acids get reassembled into triglycerides and packaged into particles called chylomicrons. And here's the detail that makes the whole anatomy click into place. Chylomicrons are too big to squeeze into a regular blood capillary. So instead, each villus has its own specialized lymphatic vessel running through its core, called a lacteal, and the chylomicrons get absorbed there instead, into the lymphatic system, not the bloodstream, traveling through lymph until the thoracic duct finally empties them into a vein near the neck. Fat, uniquely among the nutrients, skips the liver's first pass entirely and goes straight into general circulation. That's one whole mechanism running in parallel with everything happening to sugar and protein at the exact same time, in the exact same organ, and it needed its own dedicated plumbing to make it work. None of this, the segmentation contractions, the precisely timed hormone releases, the enzyme activation cascade, is being coordinated by your brain. The small intestine has its own nervous system built right into its wall, called the enteric nervous system, and it's estimated to contain about 500 million neurons, more than the entire spinal cord. Michael Gershon at Columbia University spent much of his career mapping this network in detail, and he's the one who popularized calling it the body's second brain. It can run the local mixing and moving of chyme entirely on its own, staying in touch with the brain through the vagus nerve, but perfectly capable of running the basic operation, even if that connection gets cut. You are not driving this. At best, you're being kept informed of the outcome. As fullness, as that eventual urge to defecate, as the growl of an empty migrating motor complex hours later, the actual chemistry runs on its own authority, in the dark, without ever waiting for you to notice. There's a historical figure who first proved that digestion was chemistry and not something mystical, and his experiment is genuinely uncomfortable to hear described. In 1822, a Canadian fur trapper named Alexis St. Martin was struck by a shotgun blast at close range, and the wound healed in a strange way. It left a permanent open opening straight into his stomach. The army surgeon who treated him, William Beaumont, realized he'd stumbled into an unprecedented window onto a living digestive system. Over the following years, he tied pieces of food to a string, lowered them through the opening, and pulled them back out at intervals to watch how they'd been broken down, establishing, through direct observation, that digestion was a chemical process driven by specific fluids, not just mechanical grinding or some vague, unmeasurable force. Beaumont published his findings in 1833, and they effectively founded the scientific study of gastric physiology. The chemistry we can now trace down to individual transporter proteins and named enzymes began with a gunshot wound and a piece of food tied to a string. What should really give you pause is just how narrow the tolerances are on all of this. Secretin has to fire fast enough to neutralize the acid before it damages the duodenal lining. Trypsinogen has to stay switched off until it reaches exactly the right location. Bile has to show up in enough quantity to emulsify the fat load of that specific meal, which is exactly why a very high-fat meal can outpace the whole system and leave you uncomfortable. Lactase, for a large chunk of the world's adult population, simply stops being produced in any useful quantity after childhood, because the gene that keeps it switched on past weaning isn't evenly spread across human populations. It shows up at high frequency in people whose ancestry traces back to long histories of dairy, farming, and it's largely absent everywhere else. Undigested lactose that makes it to the large intestine gets fermented by bacteria living there instead, and that's the actual mechanical cause of lactose intolerance. Not a malfunction, the expected ancestral default finally showing itself in a body that stopped needing the enzyme a long time ago. 4 to 6 hours after a typical meal, most of what can be absorbed already has been. The segmentation contractions taper off. The migrating motor complex takes over, sweeping whatever's left, fiber, unabsorbed material, slowed off cells, onward toward the large intestine, where water gets reclaimed and whatever bacteria call this system home finally get their turn with what remains. What started out as a swallowed bite has been taken apart down to the level of single molecules, sorted by type, routed through two completely separate transport systems, blood for sugar and amino acids, lymph for fat, and delivered to a body that experienced almost none of the process directly. You felt hunger. You felt satisfied once the meal was over. In between, across several hours, hormones you can't sense triggered organs you can't feel to release fluids you've never seen, all timed to a chemistry you only just found out existed. It happened the last time you ate. It's either happening again right now, or it will be again in a few hours. The tube behind your navel doesn't wait for you to understand it before it gets to work.